get started. So first, I would like to thank Prof. Chong, the director of IMS, and Prof. Sun, the director of IAS, for the great support of this our IAS and IMS joint public lecture series. So this public lecture series, for the mathematical artificial intelligence. So the main focus will be the recent progress of geometry and the topology in AI and also data. So it's my great honor and pleasure to introduce our speaker today, Professor Jürgen Jost. So Jürgen is the director of the Max Planck Institute for Mathematics since 1996. Is the member of German National Academy of Science, and his research interest is the Riemannian geometry, geometrical analysis, and the applications in complex analysis, mathematical biology, brain science, data, and recently some neural network models. He has published more than 500 papers with 19,000 citations. So I think he's one of the leaders in this area. So with that, let's welcome Jürgen to give a talk. Yeah, please go ahead.
Yeah, thank you very much for giving me the opportunity to give a presentation here. Now I will sketch you some general picture of the relation between data and mathematics with the focus on the biological sciences. But these questions are also relevant for many other fields. So first of all, we look at data where we do not know a model, where we just have certain data, and the challenge is to understand, to group, to organize these data. And for that purpose, let me first take to a small city in the countryside in Saxony in Germany, not far from Leipzig. That small town or city is called Freiberg. And in Freiberg, you have something very surprising and amazing, namely, you have the world's best collection of minerals. So the mineral collection there is superior to those that you would find in places like Paris or New York, and essentially it originated from a private donation. Now here you see some of the arrangements, and let me just show you some pictures of the minerals that you could find there. So you see an amazing variety, and you also see an amazing beauty of many of the minerals that you can find there. In fact, there's some history behind that, because the collection was already started in the 18th century by a pioneer of geology and paleontology, whose name was Abraham Werner. And Freiberg traditionally was a mining town. So in the region, of course, they could already find many minerals, but now the collection stems from all over the world. So this is just some variety of the minerals that you find there. Now imagine you either are a museum director or you are a scientist, and somehow you have to organize those. You have to group those. You have to find some structure there. Of course, they themselves are structured. You see many regularities, for instance, but how would you structure the entire collection? How would you organize it? There might be certain approaches that you can think of. You could think of physical properties. You could look at the color. You could look at the hardness, which is important. You could look at the size. You could look at the texture. You could bring them to a chemical laboratory, for example. Then you could try to analyze the chemical composition, because typically these are not pure substances, but composites. You could arrange them according to the geographical origin, that is where the different specimens have been found. You could try to organize them according to geological age, because they have been formed over millions and perhaps hundreds of millions of years. And you could typically date the stratum in which they are found, and so on. So you might come up with other ideas. So that is one typical situation, maybe where you have a collection of very rich data, but where you look for an organizing principle. Now, the scientific grouping, if you're a scientist, that uses none of the above, it uses the symmetries of the crystallographic structure, because they're typically crystals. And it was an important discovery in 1914 when the physicist Max von Laue determined such structures through X-ray diffraction. And he was in fact awarded the Nobel Prize for that achievement. Now, you know what's the structure at the atomic or molecular scale looks like, and then still you need to organize it. And it turns out that the symmetries of this structure constitute a group, which is an algebraic structure that had already been introduced in the 19th century by important mathematicians like Carl Friedrich Gauss, Évariste Galois, and Felix Klein and Sophus Lee. And the latter in particular looked at symmetry groups, and the particular groups that occur for crystallographic structures could then be classified. Now let's go a little bit into the mathematics. So a group is a very basic algebraic structure where you have a law of composition between elements. So G and H are elements, and if you have two such elements, you can form their product G times H. Certain conditions have to be satisfied. There's a neutral element, E, with which by multiplication leaves the other element invariant. So G times E is always G, and you have inverses of all elements. So for each element, you can find an inverse G to the minus 1 that when multiplied with G gives you the neutral element. And there's also an associativity rule. You can put in brackets into multiple products wherever you like, but the groups need not necessarily be commutative. So G times H is not always or not in every group equal to H times Z. And the important examples that we have here are the translations and the rotations of three-dimensional, in fact, any dimensional Euclidean space. So if you compose two translations, you get another translation. If you compose two rotations in 3D space, you get another rotation. But the rotation group of three-dimensional space already is non-commutative. The translation groups are. And you can also combine them into a single group, the group of Euclidean motion. And then the crystallographic groups and are subgroups of the groups of such Euclidean motions. And they're discrete subgroups, and you can classify them. And when you have this abstract mathematical scheme, you can order the minerals. But now let me move you to another small place, not far away from Leipzig, to Jena. And in Jena, there was a biologist who was very famous in the 19th and early 20th century, Ernst Haeckel, and in fact, he was one of the first followers of Darwin. And so he tried to relate biological species to each other. So he has a human skull and the skeleton of some monkey, and of course, he wants to point out the similarities. He was not only a biologist, he was also an accomplished artist. So he drew the biological forms that he saw and identified and put them into colorful pictures, which was very influential for the Art Nouveau, Art Deco, Jugendstil at the end of the 19th century. And as a follower of Darwin, of course, he was interested in the tree of life. Here you see his picture of a tree of life with a different species. And the essential idea is that the leaves or the tips of the branches represent the extant species, and what is in the stem represents common ancestors of the branches that emerge from there. And now let me show the picture of what I want to show you first. These are the silicon skeletons of small animals, protozoa, that he found in the sea, put under the microscope and drew, and again, he wanted to classify them. And of course, you see that these structures possess high degree of symmetry. And again, there are symmetry groups, and you could classify them according to their symmetries. And so these are very beautiful creatures. And again, very abstract mathematical concept can also be used here for data sets that is very different from the data sets that we had seen earlier. But of course, the question that you want to ask is, how does this relate to the phylogenies? That is, radiolaria with the same symmetry group or with similar symmetry groups are also phylogenetically related to each other. That is, do they have a rather recent common ancestor? Or in other words, how do these structures fit into the tree of life that Darwin also drew? And so I'll return to that, but let's first ask the general question, what is the role of mathematics for the analysis of biological and other data? We have seen some role already, that it provided abstract concepts like that of a group, which allowed us to classify symmetries. But as we shall see, also the tree of life will lead to interesting mathematical questions and structures, which then can be used to understand the data at hand better. But we will also see many other examples of mathematical structures and of biological data and of the interplay between the two. In fact, we will look at examples from population genetics, from biochemical kinetics, the first two ones only relatively briefly. Then we will again turn to phylogenies, but not only look at the fossils that Darwin and Haeckel and so on were interested in, but also at modern genetic sequences. Then we look more abstractly at network data arising from correlation patterns in biological data. And we will also look at metric data, and very briefly at the end, I give you an outlook about 3D patterns, three-dimensional patterns. So before we had data that were looking for a model, now we start with models that are looking for data. So the model is there, and then you have to ask whether the data fit or do not fit into the model. The question of population genetics is that you have individuals with different characters or traits. They recombine, reproduce, are selected, and then you will see what will happen. So on the left, you have these ants or these insects of very different colors. And in the end, on the right, you see certain processes that a group produces offspring, and then perhaps in the end, only few colors survive. So also more abstract pattern, you have a so-called source population. Then a couple of them are brought into a new environment. They're the founders of a new population. Then you have some bottlenecks. They are subjected to selected forces, and only certain ones survive, and perhaps only certain types survive. They then expand, and in the end, you have fewer types than you had in the original source population. Of course, then you can have mutations and so on, and can generate new types. So these are some of the basic ideas or concepts of evolutionary biology. And this can be turned into a mathematical model, the Wright-Fisher model, which is already almost 100 years old. And it is very mathematical. So the biology is very stylized. You have a population with carriers of different alleles, that is, alternative versions of a gene at some specified genetic loci. The simplest model, you just have one locus with two different alleles. And then in each generation, you randomly choose a couple of individuals, perhaps repeatedly, some individuals perhaps repeatedly for reproduction, and then you generate the next generation. Mutations can occur in between, and the probabilities with which individuals are chosen may depend on the individual fitness. And the entire model is described in one single equation, a partial differential equation, which is called the Fokker-Planck equation by physicists and the Kolmogorov-Forward equation by mathematicians. And it is about the probabilities that the relative frequency of one of two alleles occurs at a particular time t. And so this satisfies partial differential equation. And there's one coefficient, the drift coefficient, that incorporates all the biologically relevant effects of mutation and selection. The first part just is a random sampling of the individuals for reproduction. I will not go into the equation. I just want to show you the picture that a very general biological model with many effects can be condensed into a very simple looking mathematical equation. The mathematics behind this equation, however, is quite rich. And of course, you can and should extend it to more than two alleles, to several loci, to recombination. And from this equation and its generalization, you can compute essentially everything that interests you. For example, the expected time before one of the alleles disappears from the population. Because of these random effects, eventually, unless many mutations occur, only one allele will survive in the long term. And that we had already seen in the pictures there. In the end, you just had fewer colors represented in the population than individually. The model is very simplified. And of course, the data, the actual biological data are rather noisy and may not completely fit into the model, but nevertheless, it makes very useful predictions for plant and animal breeders. So that is some of the history. But in more recent time, it has also inspired computational optimization schemes, the so-called evolutionary and genetic algorithms. In biology, selection works and produces better and better adapted individuals. And so one also wants to use that for optimization schemes by choosing members of a population for reproduction according to how well they solve a particular task. Anyway, there's another very classical model, namely the biochemical kinetics, which also is relatively old. In particular, their enzymatic reactions. You have an enzyme E and a substrate S, and the two can bind into a complex ES, which can also be dissolved. But when you have the complex, then the substrate is transformed into a product P, and the enzyme is released again. And these reactions follow certain rules, and these rules allow you to translate the system, or the reaction system, into a system of ordinary differential equations. So I have written these four equations. So C stands for the concentration of E and S together, and E and S stand for the individual concentrations, and P, of course, is the concentration of the product P. And so S decreases by being absorbed into the complex, but it also increases by being released from the complex. likewise for the other ones. And then you can do that for as many reactions as you want, and you can then solve the resulting system of ordinary differential equations. Mathematically, this is relatively easy in principle and rather general, but of course, it ignores an important aspect, namely that the cell has a three-dimensional geometry. And here you just look at the overall concentrations. And also for large systems, if you have hundreds of reactants, the predictions of the model are often not so accurate, and remarkably, sometimes simpler approximations as Boolean approximations, that is, something is present above a certain threshold or absent, and then you have discrete dynamics can often yield better results. So there's some tension here between what the mathematical model can yield and what the biological data tell you. But let's return to the tree of life. So originally you wanted to reconstruct the tree of life on the basis of fossil findings. So you have two extant species like man and chimpanzee, and then you find some fossil that shares characteristics of both of them, and then you say this may have been the common ancestor. On the left-hand side, you see the famous sketch of such a tree by Darwin himself, the founder of evolution and biology. On the right-hand side, you see a tree as depicted by Ernst Haeckel. As I told you, he was also very talented as an artist. But just as an historical aside, such trees were not first invented by biologists. Such trees were first produced by linguists, like the 19th century Indo-Germanic linguist August Wilhelm Schleicher, and they had a similar aim. They looked at the extant languages, in this case, Indo-European languages. Deutsch is German, then you have Litauish, which is a Baltic language, then you have the Slavic languages like Russian or Polish, then you have other languages spoken formerly in Western and Southern Europe. You have Old Greek, and you have the Iranian and Indic languages. And putatively, because of the similarities that people found, and also when they could read ancient text in Greek or Indian script, then they speculated that all the Indo-European languages had derived from one common ancestor. And so that again is the principle of the tree. So the tips of the branches, as one says, the leaves, are the extant data, what you find today, and then the merging points of the branches represent putative languages or languages that have been documented in historical texts. But mostly they have been reconstructed on the basis of linguistic regularities and similarities between the extant languages. Now, how can you reconstruct a phylogenetic tree? So originally, one had the extant species, one could look at them, one could look at their so-called morphological characters, how many fingers they have, what the shape of the feet is, what the shape of particular bones is, and so on. And you also had fossil data, and you could compare them. And so on the left picture, you see something very simple. You have two extant species, A1 and A2, current species, and you have some fossil species A, and the fossil species A had some character value small a, and that character value is preserved in A1, but it has been changed to A prime in A2. Now, on the basis of this, you could develop systematic principles. So in the middle, the original character A changed to A prime in the fossil species A2, and that state then was preserved in A21, but it further changed to A double prime in A22. Or you could look at different characters. So on the right-hand side, A changes to A prime from A to A2, but there's another character whose value B is preserved in A2, but it changes to B prime from A2 to A22. And so you can easily translate that into logical principles. That is a principle of cladism by Willy Hennig in the 1960s, and that allowed for systematic analysis of the data that one had. But of course, there's the question, what is a morphological character? What you should look for? That is not so easy. Now, today, one has other types of data. One has genetic sequences of extant species. So by now, you can easily sequence the genome of almost anything. But typically, you don't have the genetic sequences of fossils, so you need to reconstruct their ancestors. So there is a basic and obvious idea. You look at the sequences and you look how different they are. And from that, you compute the distance. So the basic idea is, over evolutionary time, certain nucleotides and the genetic sequence change. And so you just record how many point mutations have occurred between two different sequences. And then this is the distance. Of course, biology is not so neat. There are also other changes. Entire regions could be deleted. Regions could be inserted. The ordering of a sequence could be inverted. There could be duplications and so on. And so that was a basic problem for bioinformatics in the beginning, but the so-called problem of sequence alignment then was solved. But even if you have such a metric, even if you have such a distance, then the data may not fit into a tree. For metric data to fit as leaves into a tree, you need to have a particular relation satisfied, the 4-point condition, which is necessary and sufficient. Let's look at some pictures. So here, the species that you have are X, Y, Z, and W. And for computing the distance, you just have to look at the length of the line segments joining them. And here, the tree condition is satisfied, so you can arrange them into a tree. And of course, then you see that Z and W are more closely related than they are to X and Y, and X and Y are also more closely related than they are to the others. So that would be a tree, and so that would visualize relationships between them. Now, here we have a different situation where the data do not really fit into a tree. So the distance between X and Y, here is the length of the segment from X to Xi, plus that from Xi to eta, plus that from eta to Y, and so on. But the distances no longer satisfy the 4-point condition. But that is what comes out of your measurement. By computing the distances between the sequence data. So what can you do there? Well, as a mathematician, you develop a theory, namely the theory of hyperconvex spaces and hyperconvex hulls, and much of it was done by my good friend Andreas Dress in close collaboration with biologists, in particular the Nobel Prize-winning biological chemist Manfred Eigen. So you fill in the region that doesn't fit into a tree, and you call that the hyperconvex hull. And so that is the best approximation of a tree that you can possibly have. It is not quite a tree, but it indicates the degree of uncertainty that you have in the data that you cannot resolve from the data and that you need to live with, that gives you the ambiguity about the phylogenetic relationship of the data. So for sequence data, we have a natural metric, the distance, that is the number of steps, mutations, and other modifications to get from one sequence to another. So that was the basis of that. And then the question was, does that fit into a tree or does it not fit into a tree, and how close is it to a tree? And that is what the mathematician then can tell the biologist. But many other data are more naturally represented as networks, and we'll come to that now. So the data points are the nodes, and two nodes are connected by an edge if there's a sufficiently high degree of interaction or correlation between the data points. So if we have such a network, how would you evaluate it? And typically, of course, the biological networks that you get from the data these days are pretty large. With hundreds, thousands, ten of thousands, hundreds of thousands or more nodes. And you cannot just look at them and detect the structure, you need methods. And in our work, we have focused on the edges because they constitute the important aspect, in our opinion, of a network. Well, let's look at a very simple edge-based quantity. So look at the picture. You have two nodes, V1 and V2, that are connected by an edge. And you want to sample or gauge the importance of an edge. And there you compute particular measures, the so-called Ritchie or Forman-Ritchie curvature of the edge. So you look at the degrees of the nodes. The degree of the node just tells you how many edges are emanating from it. So there are 4 edges emanating from V1, and there are 5 edges emanating from V2. Both, of course, include the edge E in question that we're interested in. And the Ritchie curvature of the edge then is defined as 4 minus the sum of the 2 degrees. There are some conventions behind that. So you may ask, why do we count the degrees negatively and why do we have the 4 there? Well, that comes from the history. The Ritchie curvature was first developed in a very different context, that of Riemannian geometry, founded by Bernhard Riemann and then extended by Ritchie and others in the 19th century. And you may have also seen the Ritchie curvature occurring prominently in the Einstein field equations. Now, this Ritchie curvature for edges has certain properties that are similar to the Ritchie curvatures as occurring in Riemannian geometry. I will not go into the details here, but these similarities then explain the conventions here, that you have the 4 and that you have the minus signs. Anyway, so it's a very simple quantity that you can compute very easily if you have an edge in the network, and you can then compute it for all of the edges in the network. We can look at other versions, for example, the Olivet-Ritchie curvature that also distinguishes between edges. So the 2 edges, EV1 double prime and EV2 triple prime, together form a triangle. And so, of course, the relation to the edge E is different than the other edges. If you want to look at spreading of information or diffusion or whatever through the edge, then those edges that form triangles, of course, keep things in the vicinity of E, whereas the others transport them to different parts of the network. One could, instead of looking at the sum of the degrees, also look at the difference of the degrees. We have also done that. That also yields useful measures, but I just want to indicate to you here that different measures are possible to quantify or gauge the importance of an edge, but it turns out that this basic Ritchie curvature, this Forman Ritchie curvature, already is remarkably insightful. The point is, again, that you can very easily compute it locally. For each edge, you just have to look at its vicinity in the network. You don't need to know about the entire network structure in order to compute the Ritchie curvature of a particular edge. But nevertheless, it turns out that those edges that have particularly negative Ritchie curvature are quite important, because then many shortest paths also can be expected to go through such an edge. And the distribution of the Ritchie curvatures of the edges of a network then yields a very simple approach for the quantitative evaluation of networks, and I will show you some examples. Here we look at gene co-expression data. Let me explain how the network is obtained. You have various samples, and in each sample, you check to how much each gene is expressed. You have a couple of genes, you look at the expression profiles in the various samples. They come, for example, from microarray data, which was an important technology that was introduced, whatever, maybe 20 or 25 years ago. And then you look at the correlations between the activities of different genes across the various samples. So if two genes are always expressed in the same samples and not expressed in the same samples, then the correlation is quite high. If the expression profiles of two genes are essentially independent of each other, then the correlation is very low. And of course, if one gene is typically expressed when the other gene is not expressed, and conversely, then you have a negative correlation. And so then for every pair of genes, you compute the co-expression, and then you have a matrix. And then you say, you construct a network out of that, where the genes are the nodes, and two genes are linked by an edge whenever their co-expression is above a certain threshold. Or you could have a weighted network where the weight of an edge is equal to the co-expression. So then we have a network. Of course, this is an artificial construct, but nevertheless, after all, it should tell you something important, or it should encode something important about the original data. Namely, you want to see which genes typically cooperate for achieving a particular cell type, or for achieving a particular task under a given condition in a certain cell. So then you have transformed this gene correlation data into a network. And here's an example that we analyzed. We looked at particular gene co-expression networks. We looked at the distribution of the Ritchie curvatures, and then we saw this particular pattern. And we see that there's three humps. There's one major hump, the big peak in the middle, and then there are two subsidiary humps where you have local maxima in the curvature profiles. So the data obtained for each value of the curvature, you check how many edges have that particular value, and that gives you these profiles. And then we ask, what do the humps correspond to? And we see that the major hump corresponds to a particular class of genes related to the particular task that the cell is solving at this particular point, and the measurements were made. As the smaller humps correspond to various sets of so-called housekeeping genes that are always active in the cell under every condition and for every type to maintain the basic metabolism of the cell. course, all this can be refined, but I just want to show you the pattern. Now I want to show you the same type of analysis in, again, a very different biological setting, namely brain imaging. Probably you've heard about fMRI, which stands for functional magnetic resonance imaging. And this is an imaging technique where you're put into a scanner, exposed to a magnetic field in order to measure the blood oxygen level dependent contrast. That is, how much oxygen does your blood transport to a particular region in the brain? And the idea is when the brain is active, then it needs energy, and the energy is transported by the blood. And how much energy is transported by the blood, you can detect through its oxygen level. And so then you measure that, and you see a picture where the colors indicate an increased bold level when the subject had to solve a particular task or was observed under a particular condition. Now, this is not as easy as it looks, because different people have very different brain anatomies. And so the question is, which region in one brain or which area in one brain corresponds to an area in a different brain? And so you need to pre-process the data from different individuals to align them. And for doing that, you need standardized methods. So you partition the brain into small voxels. These are cubes of 1 millimeter side length. Then you look at the activity into that voxel. I should say, of course, that the fMRI data are then taken from all directions, and through a mathematical scheme, you can then reconstruct the three-dimensional geometry. So therefore, you can partition the brain into small voxels that you then have to correlate across different individuals. You could also try to identify brain regions from anatomical atlases. So current anatomical atlases give you about 200 or 300 brain regions. But again, you need to identify them in different individuals. And of course, there are also certain physiological effects like breathing and heartbeat that also influence the blood oxygen level, and you need to subtract them. But I've been working with one of the leading people in this pre-processing of the brain imaging data, that is Gabriele Lohmann from Tübingen. Now, suppose that we have done that the data are pre-processed and aligned. How could we analyze them? Again, as I had already indicated, one will look for correlation patterns between the activities of different voxels or brain regions. And we construct a network where the voxels or the regions, whatever we want to take, are the nodes, and where the edges indicate the correlations. And how to analyze such networks? We have been working with two methods. One I will not speak about here, the spectra of Laplace operators, and the other is that which I had already highlighted and explained, the distribution of Ritchie curvatures. And that can tell us quite a lot. So here's another stylized picture from a collaboration with Arijit Samal and his group in Chennai in India. And there we compared the distribution of Ritchie curvatures between healthy subjects and other people suffering from autism. And again, we looked at the correlation networks between brain regions obtained from fMRI data, as I had explained. And you see, we can distinguish through the distribution of Ritchie curvatures quite clearly the autistic subjects corresponding to the red curve from the healthy subjects corresponding to the blue curve. So the deviations indicated in a little shading, which may, however, not be so well visible in these slides. But the point is that the difference, the ability to distinguish between them is rather clear. So in other words, in autistic subjects, the brain regions are less well connected than in healthy subjects, and that can be very easily checked through the analysis of the fMRI data by looking at the distribution of Ritchie curvatures. Here's another task which was done with Gabi Lohmann and other collaborators. And here we have been using data that were generated within the American Human Connectome project. We have then analyzed them with our methods. And the idea is the subject is put into the scanner and has to do some basic task, either a language task or working memory task. And the idea is then we measure the brain activity with fMRI and the steps that I had explained. And then we want to infer the general intelligence of that person. So in other words, we put you into a scanner for a few minutes, let you solve some simple task, and then we want to estimate your IQ, and it turns out that we can do that actually quite well by, again, using the Ritchie curvatures here. So that in these plots indicates the correlation patterns and the correlation values. And of course, other people have done that with other methods, but with our method, but the very simple evaluation of this Ritchie curvature, we get better results, that is better predictions of the general intelligence of the person in the scanner than other methods did. If you just look at the resting state and the person doesn't have to do anything, then the correlation is not so good. And that is, of course, not unexpected, because your intelligence can only be seen if you have to solve some task that requires some degree of intelligence. So again, it seems that intelligence depends on a good connection between brain regions, high correlations between activity patterns in different brain regions. Okay, let me then conclude by some general remarks. When, as a mathematician, you're confronted with data, then nowadays you are typically confronted with data where the person who generated the data doesn't really understand the meaning of the data, because in contrast to what I showed you in the beginning, these evolutionary models or the phylogenetic trees or the biochemical reaction networks, you typically no longer have a good model. The data just come because people, usually physicists, have developed some very sophisticated imaging techniques. Then, since you often do not have a guiding theory behind the data or good model behind the data, you first have to ask, what abstract properties do the data possess? Are they intrinsically low dimensional? For example, do they have only very few intrinsic degrees of freedom? Are they perhaps hierarchically organized? Are they generated by only few sources? Are they intrinsically regular and smooth, even though the actual data may be distorted by some noise? Do they have many symmetries, as did the mineral data and the radiolaria, whose examples I showed you in the beginning, and so on? Now, some data do have some of these features, but others don't. You don't know in advance. And so therefore, you need to perhaps have some good guess of what structure could be in the data, and then you apply some appropriate machine learning technique that cleverly exploits such structures. So the manifold learning, UMAP is a very good version, and then there are these autoencoders like BERT and others that check for the basic regularities and try to extract them and so on. But in order to have a general picture, you need very sophisticated mathematical theory, and that is now the challenge for mathematics, to develop an abstract theory of data, and that is also something that we are actively working on. And of course, for an outlook, I should also mention that now comes the time of deep neural networks, and there's been a spectacular application that is AlphaFold. And so the basic problem that people could not solve is to find the folding structure, the three-dimensional structure of proteins. You have the amino acid sequence that is typically 250 or 30 amino acids that protein, or more precisely a polypeptide, is composed of, that you can easily get. And that is also comes from the genetic code, because you know which triple of nucleotides codes for which amino acid. You have that amino acid sequence, but just the sequence is biologically useless. The amino acid sequence has to fold into a three-dimensional structure, and only this three-dimensional structure then lets the protein carry out its function. But the question is how to predict that. Generations of mathematicians, computer scientists, physicists have tried to develop mathematical models that can actually predict the three-dimensional structure from the sequence, but none of them really worked. The problem is extremely difficult because of the vast number of local minima of the energy landscape in the technical terminology. But AlphaFold, deep neural network, in particular AlphaFold 2, the second version from 2020, outperforms now all other schemes by a vast margin. And now we have a good scheme to predict the structure of a protein from its sequence, and we no longer have to go through the very difficult biological measurements. You have to crystalline the thing first, and then you have to use X-ray spectroscopy, as I showed you in the very beginning when the minerals were classified. You can do the same with the proteins. You can detect the structure of the proteins after crystallizing them and then shining X-rays through them and look at the diffraction patterns. But that is very time consuming. So for a single protein, that can take a PhD student a couple of years to identify the three-dimensional structure. But now we can do it computationally. But then the question is, will that also help us to make theoretical advances? AlphaFold solves the problem, solves it beautifully. Of course, still reasons, there are still possibilities for improvement. Not all the predictions are correct and so on, but it's pretty good already. But we don't understand how it does it. So the question then is, will that then also help us to make theoretical advances? Can we understand the theoretical problem better now in order to come up with a better mathematical model that then may make the prediction task easier? Okay, so with that outlook, I want to conclude. So I hope I could give you some overview of the relation between data, models, and mathematics. And the relation has been very different. In the beginning, we often had good models and we tried to fit the data into them. And then we could see to what extent the data fits the model. But now, more recently, we typically have data without underlying models obtained by various spectroscopic or imaging techniques. And we need to have abstract mathematical schemes and principles in order to analyze such data. And that, of course, also stimulates mathematics quite a lot. And then we have, of course, also very powerful computational techniques like deep neural networks to approach the data, but again, that poses important challenges for mathematics. So thank you very much for your attention, and of course, I should be happy to try to answer any questions that you may have.
Thank you. Any questions from the audience? I think one people just type in the chat and
Okay.
I can say it.
yeah, maybe you can directly.
Hi. My name is Eric. I used to work as a machine learning consultant in a company, and I noticed that in the case of machine learning, people have a lot of, or also in statistics, people have a lot of theory about what kind of properties should a data set have to be convenient, to have low bias. But in reality, companies cannot afford to produce such a nice data set to solve a problem. An example is a big company such as Google. They produce, they open this service, a dermatology app, that doesn't work with people with darker skin. And so if Google cannot afford to create a data set that has, that can work for everybody, smaller companies like startups doesn't have the money to produce such a good data set. So I wonder if there is some interest of the math community to, try to find a way to produce data cheaply. Like, yeah, we know what the data should have in order to be useful, but we cannot spend money to create such data sets. So what will be something practical? What will be something that is actually useful for companies? And I don't know if people is aware of these problems, but we face it in a startup, and I think it will be interesting to see if there is people working on a cheap way to solve realistically to create data sets.
Yeah, thank you. Thank you for pointing this out. I don't think there's an easy answer for that. On one hand, of course, in certain cases, you do have the data. That is the area of big data. We don't have that many big data in biology, but in other fields, in physics, social sciences, astronomy or so, we really have gigantic data sets. And then the question is how to handle them. But that also requires then a lot of computing facilities. In other cases, of course, you may not have the data, as you point out. Now, of course, many data in other fields are extracted from the internet. Yeah, you have lots of pictures, images, whatever, text and so on, and you can extract and download a lot of that from the internet. So the big companies have an advantage because they have the resources to extract, download, and store all those data. In other cases, of course, as you point out, you do not have the data, but you want to test the methods. Then I think the only realistic way is to work with simulated data. That has some advantages and disadvantages. The disadvantages, obviously, that the data are not real data, so they may contain artifacts based on the way that you have generated them that do not correspond to anything in reality. But on the other hand, you can relatively easily produce them, and you can also vary them by changing certain parameters in the data generation process. There are certain characteristics of real data that you can also check. So many real data have certain power law type of statistics. So there is a important zip law, for example, that applies to many social and infrastructural data. And when you simulate your data, you should check that such general statistical features are preserved in the data set. The financial data analysts call them so-called stylized facts. And they also have such problems that often they do not have the data. The data are of course valuable and proprietary, and so you do not get them so easily for free. And then again, you need to test your models with simulated data, but then the simulated data should at least represent those general statistical facts, what these people call stylized facts, that have been consistently found in real data. But so the general way forward, of course, would be to try to look for simulated data, but then you need to establish criteria in order to check that the simulated data contain all the important, obvious, or perhaps hidden features that real data have.
Thank you.
Any other questions? You can just unmute and ask. Yeah.
Professor, there are questions in the chat window.
So yes, so
Yeah, then I should stop sharing my screen, then I can see the chat window.
I think ideally, if you want, you can just unmute and ask yourself.
Yeah, sure. Thank you. So the brain imaging research, if it is showing that IQ in otherwise physiologically / neurologically normal individuals is strongly predicted by the brain structure, is that then some evidence for the objectivity of IQ tests?
I was just kicked out for a moment, but I hope that you can see and hear me now again.
Yes, so maybe Adele, you can ask again. Yeah, sorry about that. Yeah.
Yeah, sure, no problem. So the brain imaging research, if they're showing that IQ in otherwise physiologically or neurologically normal individuals is strongly predicted by brain structure, then is that some evidence for the objectivity of IQ tests?
Yes, yes, that definitely is a very, very important question. I mean, the validity of the IQ as a general measure of intelligence, of course, has been questioned. And perhaps it's not the general idea of the IQ, but perhaps certain components of it. And our brain imaging data show that certain components of the IQ, as measured by the psychological tests, correlate better with what we see in the brain images than others. And so that is something that we should then discuss with the psychologists, whether they perhaps should give more weight to those components of the IQ that better correlate with our data, or whether there's something in the IQ, certain aspects of the IQ that cannot be so easily detected from brain imaging measurements. So that should be an important debate in the near future. But I don't know the answer.
Thank you.
There's one more question in the chat by Kang Rick. He's asking about, based on today's lecture content, how will modeling help predict potential adversary attacks such as cyber attacks?
Okay, there's something there, because with these network measures, you can also try to gauge the vulnerability of a network, yeah, and in particular, identify those edges that are particularly important for the functioning of the network, and then try to protect those. In particular, those edges that have a particularly negative Ritchie curvature in general should be rather important for the network. And so you could try to protect those or duplicate those, or or provide alternatives to them in the network to make the network more resilient. And so there are also other mathematical approaches to this. We have developed certain complexity measures that correspond to the resilience or robustness of the network by estimating its ability to reconfigure itself after a random attack. Now, that is not a targeted attack. In the beginning, I talked about targeted attacks, but if you have a random attack, then the network can protect itself against that by generating a high degree of complexity, that is interrelations between different parts of the network. We can also quantify that.
Maybe I can use this on teachers to ask questions. So in your models, you mentioned about like a discrete Forman rich culture, other discrete versions. Usually the geometric models are in a continuous way, and then to have these discrete models on like graph or simplicial complex, what is usually the most big trouble or challenges, and where other important geometrical concepts and models can be discretized and then further used in the study?
Yeah, well, I mean, the data, usually for us, the problem is a little different. The data that we get are discrete, but we assume that the underlying structure is a continuous one. And we want to reconstruct that underlying continuous structure by simply by including more and more samples, that is by making the network denser and denser. Then there are mathematical theorems about that in manifold learning and so on, that we are also working on to estimate or mathematically justify going to a continuum limit. Yeah, and then we need to check which properties of the network actually then converge to the corresponding properties of the underlying continuous structure. And there, the spectra of graph Laplacians are quite important. The eigenvalues and the eigenfunctions of graph Laplacians are important tools. And then with modern mathematical methods, we can then prove convergence results there. And so that is a very important issue that you are addressing, the relation between discrete data and underlying continuous structures. The other way, of course, also occurs if you have a continuous structure, as in fluid dynamics or so. For computational reasons, you need to discretize it.
Yes.
Yeah, but there are also well-established schemes in numerical analysis, for instance, for that, and people then also have good convergence results.
Yeah, thank you. Yeah, any other questions? If not, let's thank Jürgen again. Yeah, fantastic talk. Thank you. Yeah. Yeah, I think that's all.
Thank you.
Yeah.
Thank you very much, and have a good evening.